Hi everyone. This lab is about sterile technique. So we're going to learn just about the theory behind sterile technique. Sterile technique is a very important method that all scientists, not just microbiologists, use. So in clinical labs, meaning hospital labs or research labs, when scientists run experiments, we have to make sure that we're not contaminating our experiments and our bacteria that you're working with. And that's basically all sterile technique is. It's a method that came about to teach people that you should make sure that you are not contaminating anything. So the technique allows you to transfer microbes from one place to another without contamination. That's what sterile technique is, and that's the purpose. Is how if you have like bacteria in a test tube here, and I want to transfer it to an agar plate, how do I transfer it, making sure that I only get that bacteria and nothing else? And that's what sterile technique. It's transferring microbes without any contamination. Most cultures we use in lab are pure cultures. I want you to keep that in mind. Pure cultures means it only contains one organism. So if we say that this is a pure E. coli culture, it means that there's only the E. coli bacteria in it. There's no H. pylori, there's no salmonella, there's no other bacteria. Mixed culture means that there's a lot of different bacteria. And so if you think about it, if you take a sample, maybe you have someone that has a sore throat, and you take a sample from their throat and you want to culture it, meaning you want to grow that bacteria, the potential bacteria that they have an infection with, you want to make sure that you're not getting any other bacteria. And that's exactly what sterile technique is. So there is contamination everywhere. There's contamination on your lab bench. There's bacteria on our hands, there's microbes everywhere, there's fungal spores everywhere. So we want to make sure that we always use sterile technique when we're running experiments. And sterile technique prevents contamination. It prevents contamination for the purposes of this class of two things: of your source culture, meaning the bacteria. When I say culture, you'll learn about this later, I mean the bacteria you're working with, and whatever media, or media means where you grow your bacteria on, that media. So if I have bacteria like E. coli bacteria and I want to grow it on an agar plate, that's my medium. I want to make sure that the bacteria does not get contaminated and my agar plate doesn't get contaminated. So that's the whole point of sterile technique. And I'll talk about what we do to use sterile technique just after a little bit more background. So sterile technique is in lab. In the hospital setting or healthcare setting, we say aseptic technique. So doctors and all healthcare staff, nurses, use aseptic technique or asepsis to prevent any contamination between patients and health personnel. So patients are in the hospital because they're obviously sick, they're dealing with something, and then health personnel, the doctors, the nurses, the x-ray technicians, everyone working in the hospital, they also harbor a lot of their own microbes. So doctors and nurses and everyone else, they make sure that they're very clean. They're wearing gloves, they're wearing PPE, personal protective equipment. They have their lab coats on to not contaminate other patients and also to not contaminate themselves. So that's aseptic technique. And we will learn about later about something called hospital acquired infections. I want everyone to keep in mind that between 5 to 15% of hospitalized patients acquire healthcare associated infections. These are infections that could be completely taken away if hospitals really did aseptic technique well. And the problem is that things are moving so quickly, and sometimes people don't have time to take the extra step to make sure that they're doing aseptic technique. They're washing their hands between patients, all these things. So hospital acquired infections, which are also called nosocomial infections, you'll learn about them later in lecture, are a very big problem we have. And the way to get rid of a lot of these hospital acquired infections or to decrease this is with really good aseptic technique. So aseptic technique is for hospital environments, and then for in lab we use sterile technique. So sterile technique involves a few things. It basically, the main goal of sterile technique is that you do not contaminate yourself and you do not contaminate the experiment you're running. So the first thing we do in lab is you always wipe your lab bench, and I've done this a lot as a scientist. Before you run any experiment, you wipe down your lab bench with some disinfecting solution, Lysol, alcohol, anything like that that will disinfect. Then you wash your hands. You want to make sure that when you're running any experiment, you're washing your hands really well, because our hands have a lot of bacteria, a lot of microbes on them, and we don't want to ruin the experiment we're running. Now, another important point is that this is a microbiology class, so you transfer back. I know this is an online class right now, but in the lab, you're transferring bacteria a lot. So to transfer bacteria, we use something called an inoculating loop. Here's a little image of it. So this person is holding the inoculating loop. And before you grab any bacteria, you always want to heat sterilize it. So anytime we're working with anything in lab, we want to pass it through the flame if it won't break, so that we sterilize it, because heat kills most microbes. So you sterilize it. And also, when you have a test tube, you pass the mouth of it over the flame when you open it and when you close it. The reason we do this is it creates a heat current or convection current. Therefore, all the airborne microbes cannot get in your culture. And I have a video where I did I demonstrated this with another technique, so you'll see me doing sterile technique. So keep that in mind. And I always ask this to my students: when you're working with a culture, do you set the cap of the tube on your desk? And the answer should be no. You want to hold everything. You want to minimize contaminating the material you're working with as much as possible. So that's the sterile technique. And the main things I want you to get is know the steps in sterile technique. So know how you have to wash your hands, you have to wipe your bench, you have to always pass your inoculating loop or anything you're grabbing bacteria with over the flame. Make sure that your test tube you pass over the flame to create a current. Aseptic technique is the name of the technique that's used in hospitals, and it's very important to limit as much as we can infections. And know why we perform all of these, whether it's sterile technique in lab or aseptic technique in the hospital. We're trying to limit contamination. So that's the goal with all of this.